Well, good afternoon, everybody. I think it's just about the afternoon. And welcome to the DRO Dementia Seminar Series. We're delighted that you can join us. My name is John Gallacher. I'm Director of Dementia's Platform UK. And it's my absolute pleasure to welcome Professor Escott-Price from Cardiff University. Now, Valentina is a very able statistician, I should say mathematician, who has applied her, how can I say this? Numerical ability, largely to polygenic risk scores, but there's much more to her than this. And we are looking forward very much, Valentina, to hear what you have to say. So over to you.
Yes, thank you, John, so much. Yes, I am a data analyst. I'm a mathematician by training, and I work in the Department of Mental Health and Brain Disorders at Cardiff University. I do work for age-related diseases and disorders, and also I work for neurodevelopmental diseases and disorders, which is schizophrenia, bipolar disorder, etc. I will be talking today mostly about Alzheimer's disease and also in genetics of neurodegenerative disorders, because that is my current interest. But the conclusions which you will see perhaps or agree or disagree with me at the end of my presentation actually is based on my expertise on analyzing both data sets, or both types of disorders, neurodevelopmental, they have their own issues and aspects, and neurodegenerative. And today I'll be talking about genetic architecture of Alzheimer's disease and why, how to predict Alzheimer's disease, risk of Alzheimer's disease, and why it works and why it does not work, and how it's related to age. So, as you know that dementia, let's start with a broader definition of dementia is primarily disease of aging, and the prevalence of dementia increases with age. Everybody knows that. I will be talking about Alzheimer's disease because it's the most common form of dementia. About 60% of cases with dementia have diagnosed with Alzheimer's disease, and it's mostly studied. So the largest data is available for Alzheimer's disease, less so for other type of dementias. What is interesting about Alzheimer's disease, that the pathologic definition of both disease and controls is different at different ages. It's also known fact. It's a very clear pathologic separation between cases and controls when they are below 65 on the brain scans, and not so clear separation when they are at age 19 between cases and controls with Alzheimer's disease. Also, another facts which are known, and the paper which we recently published, but it's known before we published it, we looked at a SEO database, which is clinical health records of Wales, covering about 20 years of the population of Wales with all clinical records. And we looked at incidence and prevalence of Alzheimer's disease and other dementias, in fact, in Welsh population. And in the first picture here, you can see that incidence and prevalence of Alzheimer's disease, and that is different curves represent different age groups, is increasing with age, with time in all the age groups. It's 90 to 80 and 70 as well. But here it's not as clear. When you zoom into this picture, you can see that in fact in younger age groups it's slightly going down, but in older age groups it's going down. The same reflects with the prevalence. So it's not only attributed to the fact that people are living longer, we are counting for the fact that people are living longer. And the main message of this observation is that a mean age of diagnosis of dementia is increased. So people are being diagnosed later, perhaps because they have better lifestyle and they don't expose the symptoms of dementia and Alzheimer's disease until later age. And also new cases of dementia risen, and people with dementia is also living longer. So all this, it brings that incidence and prevalence growth in the population. Now, since we're talking about genetics, we should slightly touch upon heritability of Alzheimer's disease. And from twin studies, is kind of agreed to be between 69 to 80% heritability, broad sense heritability. So accounting for the environment, accounting for shared environment between twins, and accounting for genetics. I will be talking about SNP-based or genetic heritability in a second. What also important for my talk and for you to remember that individual have the same genetic volume for Alzheimer's disease at any age. However, as I've shown in the previous slide, prevalence of Alzheimer's disease is dependent on age. So the higher we look at population, the higher age of population we look at, the higher prevalence of the disease is. Now let's take a look in details about heritability of Alzheimer's disease. So we know that about 60 to 80%, as I said, is genetic from broad sense heritability from twin studies, and the other 35% believed to be modifiable and environmental. Now let's look at genetics only. So the more than 1% of heritability is attributed to familiar early onset of Alzheimer's disease. We know that ApoE is the highest risk factor for late onset of Alzheimer's disease, and it accounts about 5 to 9% heritability from different studies. We knew that genome-wide significant loss of smaller effect, however, the genome-wide significant account for another 3 to 8%. And we know that there is a big chunk of other SNPs heritability, which is accounting for other 24 to 53%. There is a bit of other genetic variation which we didn't capture with genome-wide association studies, and this is a rare variance, at least at this stage, they are not well studied yet. It's interactions between SNPs, so etc., etc. So there are some other genetic heritability which we don't know how it works and how it contributes to the whole heritability picture. Looking at the history, a little bit of the history of genome-wide association studies. So the first one was published in 2010-2009. And the first, which I'm showing here, it's a pathologically confirmed, relatively small number of cases and controls. And heritability estimate is about 40%. So look, if you remember my previous picture, it's about what the green color was accounting for. The next study was from the Cardiff University and several more from our colleagues, and we looked at a larger number of people with late onset of Alzheimer's disease, and heritability estimates were 17% reported in Harold et al., and then not reported in the other two studies at that time, because the software didn't exist, to be honest, to estimate heritability, SNP-based heritability properly. Then in the other study, again, it was Nudge et al. They estimated 25% heritability. And as you can see, the number of cases and controls, the sample size is growing up to 1 million and 1.1 million, and heritability estimates are dropping. And that is actually quite a surprise. And people could not explain why that happens. One explanation, potentially, if you look at age here, what I put here. So in earlier studies where we have high percent heritability, they were past confirmed, pathologically confirmed, clear diagnosis, people of older age. Then we started including boosting numbers, and we started including cases, controls from population. So those people who might develop neurodegenerative disease or Alzheimer's disease later, but have not developed yet. So we started contaminating samples. We started taking clinical diagnosis that might influence heritability estimates. However, of course, another possibility, it's a software which has been developed to do so. So if the first studies were using GCTA software, here we have, it's the same basically what was in the table, but looking at the heritability, you see up to 40% in the earlier studies and going down to 3%, 4% in the later studies. And you can argue with me that that was estimated with LD score regression so basically using summary statistics. So I went thinking about this, I went and investigated it in the data sets available to me, looking at individual genotypes, throw genotypes, and using GCTA software for all studies which I had access to. So the first one, A here, and also picture A there, it's a amyloid confirmed or not confirmed cases and controls. You can see age of them was 65, 62. And that is a heritability estimate. So on the y-axis, it's heritability which we pick up with, and on the x-axis, a number of different methods and approaches. So if the first three looks at all SNPs taken together, the next three bars, it's looking at all SNPs excluding APOE region, and the last one is at all SNPs excluding APOE region and genome-wide significant SNPs. And the three bars within the section corresponds to different disease prevalence. So if you set up 2% lifetime prevalence, that's a heritability estimates about 35% or over 40% here. Then if you look at thinking of older age, about 5% prevalence in the population, older population is higher, and it's even higher estimates if you assume that the prevalence of the disease of 15%, which are in the age 70 plus. And you can see in the first stage study, which well characterized cases and controls, we see about 40% heritability, the same as in pathologically confirmed study, that's the one, and is the same as in Rose map study, that's the one. So you can see similar pattern here. Now, that is interesting study, Grace, where the cases were 79 years old, so they had chance to develop Alzheimer's disease and were diagnosed with that clinically. However, controls were 54 years old taken from a population. And you see heritability estimates, when we don't match cases and controls by age, drops dramatically for whatever scenario we take. Similar thing also applies to the study. It's the same Danish study, but here cases were clinically assessed without amyloid confirmation. And you can see if the diagnosis is not as good, controls are the same for both studies. If the diagnosis is not as clear, the heritability estimate drops. And finally, this is the UK Biobank data. And we've taken all people who already diagnosed with Alzheimer's disease and randomly selected 10,000 controls for calculation purposes. What we can see only when we include ApoE4 status, we see some heritability, decent heritability estimate 30–40%. And if we exclude ApoE, it's not there. So something is relatively interesting going on here. And what we know about UK Biobank, it is relatively young sample. So again, we have cases who already developed Alzheimer's disease at earlier age, 60, 65, 68, and controls are still have not developed it, being perhaps too young to, or not given enough chance or time to manifest the disease. So it's definitely something going on with age there. And thinking about this, I found this paper which showing simply ApoE allele frequencies in a population, not cases controls, population. So ApoE4 allele frequency going down with age. On x-axis we've got age. Is ApoE3 allele frequencies goes up with age. It's not like people have more E3 allele, so simply people who develop Alzheimer's disease younger, they live in average 5–7 years after the development of the disease, and therefore people with E4 alleles, they basically die out, and frequency of E4 in all the groups are smaller. However, as you remember my prevalence and incidence pictures, the prevalence of the disease going up with age. There is a contradiction here, which is interesting for mathematicians to work with. And this is a line, it's E2 allele frequency, so it's kind of quite minor and remains, well, slightly goes up perhaps with age. So why is that? As I've said already, in the different paper, it has been shown that if you're ApoE4 homozygous, then you develop your disease at about 68, in average 68 years of age. And if you're non-carrier, it's 84 years of age. So 16 years difference between disease development manifesting the disease. And therefore, genetic architecture is also relevant to the age of the population which you're looking at. So far, I have shown you facts which are known from the literature, and in conclusion, why I think that the disease architecture of genetic depends on age for Alzheimer's disease, because when we have early AD cases, about 30 to 50 years old, we know that they are attributed for familiar and highly penetrant mutations, and you don't need a genome-wide study, you don't need anything. that's only gene. If you have it, that's it. E44 carriers aged onset 60 years of age, and the prevalence is 2-3% in the population, and that's what we see from population studies. And E4 non-carriers aged onset 84 years of age, they develop disease later, and the prevalence of the disease, it's a majority of people, it grows in an older age. So conclusion so far, genetic architecture of Alzheimer's disease is different depending on clinical age at onset. And ApoE4 is not only the risk gene for AD, but it's also a disease modifier because it makes people have its onset earlier. Now, polygenic risk scores, that's what John mentioned, and I started working with in 2015. So as our first paper was published looking at 3,000 cases and 1,500 controls, and we observed that if you E4 carrier, so we calculated area under the curve here using E4 alleles, then with genome-wide significant SNPs, 20 it was at that time, and then we started including more SNPs which are not genome-wide significant, but showing some evidence, statistical evidence for association. And what we observed that the area under the curve of sensitivity and specificity was the highest when we included about 68,000 SNPs, additional SNPs to the genome-wide significant ones, so showing that that's kind of predictive. And also it was statistically significant, improving the model over and above ApoE and genome-wide significant SNPs. So that's what we have found. However, people were trying to replicate our results, and that is our polygenic model. So we include not only genome-wide significant SNPs, but something with a lower significance level up to here in the orange box, and people could not replicate that results in their studies. So I was criticized, and obviously as a mathematician, I had to resolve the problem. So people came up, Zhang et al. 2020, with oligogenic model, suggesting that it's not many or thousands of SNPs contributing to a disease, but only genome-wide significant SNPs contributing to a disease. So for this particular model, they suggest that it's about 90 SNPs which should be used, and that's it. I had to resolve the paradox because it was interesting for me. So what we've done then, I looking at the age, I was coming back thinking what could be different between samples which we analyzed and used. So we looked at independent sample, and that is a combination of ADNI and ROSMAP AAD&P sample. And we looked again at allele frequencies and polygenic risk score values depending on age of people in that sample. So if you look again here at if E2, E3, and E4 allele frequencies, E3 goes up, as in the previous study we've seen earlier. E4 goes down with age, because people developing disease earlier and die, and this is why when they are older, the frequency of E4 in that particular older sample going down, and E2 slightly going up. So that was fine, amazing. Now we split the same sample into cases and controls. ApoE4 allele frequencies were higher in cases and in controls as we expected. They were going down with age, as in the whole sample as we expected. They were also going down with age in controls, obviously, because ApoE4 also associated with other comorbidities, with other old age diseases. And these are our polygenic and oligogenic risk scores. So green one is oligogenic score based upon genome-wide significant SNPs. It's going down the same as ApoE, but polygenic risk score, when we included ApoE region, was going up with age. So that was showing to us that perhaps polygenic component is more predictive when we're looking at people at older age. And indeed, when we have taken a data set and split it, not the big values here, but it was independent from the GWAS which we used, and split it by age below 80 and above 80, what we observed, area under the curve was higher, predicted by ApoE only in a younger group. It was still predictive by polygenic component, excluding ApoE region in this group, but lower, and overall area under the curve prediction accuracy was 80% when we just put it together. Now, when we look at all the sample, look, now AUC, as expected, area under the curve dropped to 66%. However, polygenic component was, excluding ApoE again, was predicting better than ApoE. And again, when we combined it together, the two, it was about 80% the same. So what it tells us, in order to properly, in Alzheimer's disease in particular, predict risk of Alzheimer's disease, we need to model the genetic component in two ways. ApoE is a single variable, and polygenic risk score is another variable without ApoE region. And in this way, we achieve adequate accuracy in both younger groups and in older groups, because in younger groups, ApoE is more predictive. In older group, polygenic risk score is more predictive, and we combine it together, and that's what happens. So another thing, perhaps quickly to show you, I like this picture. It's explained to me why we don't, my picture, theoretical picture, if you like, why we are not predictive, why we in genome-wide association studies see sometimes opposite direction of effect size. So this is the real stuff. So we have controls, and it's again allele frequency with age. I just taken numbers from the literature, and I plotted allele frequencies with age going down with ApoE allele. In cases and going, it's higher to start with and going down quicker than in controls. And now imagine genome-wide significance SNPs with small effect size. So the difference between them is not that high to start with. And obviously, if it goes down in the same way, you see odds ratio in a younger sample could be greater than 1. It could show odds ratio equal to 1, or it can even have a reverse effect size, depending on age. This is picture, okay, that is a hypothetical picture which I plotted based upon ApoE behavior. However, I've seen it in practice for some SNPs. So that's what happens, why we don't see the same direction of effect size if we don't look at the specific age groups, because the disease architecture, genetic disease architecture is different. So with PRS now, as you know, there are tons of approaches. So at the early stage, we use just pruning and thresholding approach. Now there is tons of approaches to calculate polygenic risk scores. So pruning and thresholding, PRS-CS methods, which is a Bayesian regression framework, and also quick PRS methods, which combines LDAC and SBIRS-R. Well, who knows these methods? They know what I'm talking about, who don't just ignore just. There are many methods which we try to improve over the standard approach. And I was quite often asked question, which method to use? Does it account properly for this genetic disease architecture? Would that account for this ApoE4 effect? So we've done, again, the analysis in 2 independent data sets. Again, we made them independent from the genome-wide association study, which we used to calculate polygenic risk score, and the biofinder is completely independent, and that's data we had in collaboration with Eisai. So they were analyzing data, Eisai pharmaceutical company, and we were just basically asking them to replicate what we are doing. So you can see not too large numbers of cases and controls, but independent from GWAS. Age distribution pretty much the same. Sex distribution and ApoE carrier distribution is the same between 2 samples. So that was good to look at. So then we again had tested 5 models, predicting with ApoE only, predicting with ApoE region, predicting with PRS, as people who don't understand the disease architecture would do. Just put the whole genome into the method, get the PRS value, do the area under the curve. So that's one. Then we also did excluding ApoE region, and then we also did it using our model when we modeled 2 variables separately, ApoE separate variable and the PRS without ApoE as a separate variable. I know that it's lots of analysis here. However, what to take the message, so that is each block is a different approach, pruning and thresholding, PRS-CS, PRS-CS using UK Biobank as a reference panel, and quick PRS, it's a combination of LDAC and SBIRS-R. Basically, we covered the whole field of PRS, possible PRS calculations. And this dark green bar everywhere, it's ApoE only. Then we excluded ApoE. This orange bar, it's when you calculate PRS simply without thinking, using software and trying to predict. You see, it's not as good as ApoE sometimes. And this is a dark red bar is what our model, which we suggest 2 variables together. So the take-home message is, whatever method we use, the approach which we suggest, ApoE plus PRS excluding ApoE, is the best but similar across across different approaches. So we're achieving pretty much the same accuracy, which is good. You can say use any method you like, but think about disease architecture before using ApoE, before using PRS predicting your disease status. ApoE alone predicts good, and it predicts better. That's first bars using I think Canco summary statistics, and these bars are using the latest GWAS, which includes UK Biobank, etc., and proxy cases, basically by family history. So, but anyway, the main conclusion from all this is that model two variables to predict Alzheimer's disease, it will account for age distribution in your sample, and don't use it just a method as orange bar. You see, it doesn't behave as good as if you use the proper approach. However, it's not the end of the story. There is more to that. When comparing, so prediction accuracy is okay, pretty much the same across methods, but other individual scores of people are the same across approaches, and they're not. So using one approach, a person could appear with a high polygenic risk score, but when you use a different approach or different summary statistic, a person could be in the middle, the same person could be in the middle of distribution of polygenic risk scores. And that's again something we need to know about, not be surprised, because that's real life. And these tables basically show correlations or a percentage of overlap between polygenic extremes. You see only 40%, 50%, 49% overlap between them, depending on methodologies. Also, we try to do it using different approaches and also using different summary statistics. So the same disease, summary statistics, and even then, we don't see perfect overlap, even with the same method. Right, so be careful when you calculate PRSs and how you interpret them. I can tell you details how to interpret them later. Also, that's another picture demonstrating basically the same effect. Again, we've taken different approaches here to calculate polygenic risk scores, and then we looked at methods overlap. So the most overlapping method, provenance thresholding, then precise is a software, which is similar to that PRS-CS. So these methods seem to be more consistently predicting the same people with high or low PRS when we use different methodological approaches. Another thing which probably important to mention, biomarkers, how biomarkers predict. Perhaps genetic is not so precise. Maybe we should use biomarkers. Maybe they're better predictors. So we again published paper and investigated prediction accuracy of five biomarkers, AB40, AB42, and I said 6 in the beginning because we looked at also it's a ratio of the two. And these biomarkers, they look, they measure free amyloid beta proteins of chain 40 and 42, GFAP or glial fibrillary acid protein, looking at astrocyte proliferation, NFL, neurofilament light, it's a marker of neurodegeneration and neuronal integrity, and p-tau, and that is a marker of tau pathophysiology and amyloid plaque deposition. So these five biomarkers, 6 variables we investigated for prediction of Alzheimer's disease and PRS as well. You will see the results. So what is known about biomarkers? Yes, it's known that these AB40 and AB42 reduced in AD cases as compared to MCIs and healthy controls, that GFAP and NFL levels and CSF in plasma higher in AD and correlate with cognitive impairment. And we also know that plasma p-tau predicts Alzheimer's disease pathology 8 years before autopsy. So basically, that's what we knew about from the literature. So looking at biomarkers over time or with age, as AD pathology progresses, biomarkers for those pathologies reflect this. Some AD pathologies also seen in normal aging people at lesser rates. And also abnormal changes in biomarkers in healthy individuals may indicate incipient Alzheimer's disease before clinical assessment. Right, so that's what we knew. That's our sample. We had cases and controls. Now look here how interesting it was. So we looked at cases at 68 years of age, and controls were older. You can say good or bad. But so controls probably are more likely to be controls because they had chance to develop Alzheimer's disease. And cases are definitely cases that were clinically assessed. So we had in total about 2,000 people, 1,500 cases, 500 totally screen controls. And look at the behavior of biomarkers, again, depending on age. So on x-axis, we have age of these people, and on y-axis, we have a biomarker value. So the dark red line corresponds to cases, and you can see that our cases were, and these are controls. So you can see that, so cases, controls 82, yes. You can see that controls are systematically older, so the line is shorter. But you can see that in both in cases and in controls, biomarkers behavior is similar. So it's also increases in cases and in controls with age, and that was for AB40, similar for AB42, similar for all biomarkers. So even if you are a healthy control, your biomarkers change with age. Yes, we need to adjust for age when we analyze our data. But imagine if our cases were taken from this age, well, it's a bad example. So if our controls from here and cases are from there, you could see opposite effect size. Again, we need to be careful what age we are analyzing, what conclusions we are drawing from our results. So from these pictures, you can see that young cases quite often look like elderly controls, and also age is a significant predictor of biomarker levels in both cases and controls. So now, if you look at cases only, we looked at age at onset, association with biomarkers, and disease duration with biomarkers, and you can see all of them are significant. All biomarkers are significant predictors of age at onset. And for disease duration, the most significant predictors are GFAP and NFL, biomarkers of neurodegeneration. So next. Okay, so I wanted to say a bit more here. So what I wanted you to convince about that biomarkers behavior and genetics polygenic risk score behavior depends on age. And diagnostic accuracy for Alzheimer's disease is still not perfect. Genome-wide association studies provide less reliable estimates as they are getting bigger, providing a smaller fraction of explained heritability. Genetic architecture is different depending on age of clinical onset. Since Alzheimer's disease is a very common cause of age-related mortality, choice of age-matched controls is essential. Plasma biomarkers can only be used as a tool to predict Alzheimer's disease in the context of other genetic information, and we need longitudinal studies to look at that. What I was missing, obviously, I missed one slide to show you, when we combine together polygenic risk scores and biomarkers for the same study. So overall, biomarkers predicted, taken all together, with accuracy about 69–70%. As you've seen before, genetic has predicted, in average, about 70%, unless the samples are really split by age and analyzed properly. But in this sample, it was also 70%. When we combine together biomarkers and genetics, the prediction accuracy went over 80%. So the last one is a conclusion, which I would like to make, that we need biomarkers and genetics taken together to predict cases and controls. And moreover, we need to have brain imaging and pathology confirmation to actually proper clinical diagnosis to assess how good our predictors are in terms of disease risk prediction. So I will finish here. And of course, we did not do it alone. We did it in collaboration. Well, first of all, my postdocs, my team, who helped me to do the data analysis, and people who were generating biomarkers. It's Henrik and Amanda, and people who were helping me with have funds. Yeah, so that's, I'll take questions.
Okay, Valentina, thank you very much indeed. Would you mind putting the conclusion slide back up? I think that would just help people to focus their questions. Excellent, excellent. Well, whilst people are thinking of a question, here's a starter for 10. I can understand fully how as the allele frequency will change with age. You explained it very well. People not only dying from Alzheimer's disease, but other comorbidities. So I can understand how there is a change in the relative impact of ApoE4 and PRS scores. Okay. What I'm not clear about is whether there is actually a change in the PRS algorithm with age, or whether it's the same algorithm having a differential effect with age.
So the algorithm is to understand what contributes to early onset and to what contributes to late onset, split. So the mathematics to calculate PRS is the same, but the SNPs which go into the PRS are different, depending on age.
Yeah. Okay, lovely. Thank you.
That's what I would say, yes.
That's really clear. Thank you. Ellen, would you like to ask your question?
Sorry, can you hear me?
Yeah.
Yes.
Oh, okay. Just relating to the grey box, I wasn't sure why larger GUSs provided less reliable estimates, because I would just think if it's larger, bigger sample size, more.
Yes, that is what is always believed. And that's again, as a mathematician, I will tell you, not always. So what happens? Imagine we have just simple example, two samples. One is young age, which is ApoE4 is more predictive, and ApoE-related SNPs, so biological processes, are more predictive. And then we have an older sample, which some other biological processes are disrupted, and other SNPs are more predictive. So in one sample, we have high effect sizes for ApoE-related pathways. In the older sample, we will have higher effect sizes in ApoE non-related pathways. Now we sum it together, we put it into one sample, and then we don't have high effects for either of these. So although standard error for estimates reduces because sample size is larger, but effect sizes are also masked. And therefore, it's just, in average, it's okay, but it's not perfect. Does it make sense how I explained it?
Yes, thank you so much. Thank you.
Yeah.
Okay. So, Valentina, this displays my statistical ignorance. Okay. So, if you're looking at an effect of age and you control for age, are you not over-controlling?
Yes, you might be. You might be. So some studies, if you notice, some studies adjust for age, being conservative, and some studies do not adjust for age but then they are criticized for being, for being not so conservative. But again, in my example, if you look at this one, it was actually quite interesting for me to, for this sample. Look, we have cases and controls here, and cases are younger. So if you simply run association analysis of AD status versus age, you will find that younger age is risk factor for Alzheimer's disease, simply because of sample construction is like this. And then if we adjust for it when we're looking at the biomarkers, we will be over conservative. This is why we've done analysis in cases separately, in controls separately, shown these differences, and then did some statistical tricks to adjust for age rather than simply including it as a covariate. Yes, it is interesting.
Yeah, no, I genuinely so. I think analysis of change is absolutely interesting. Okay, well, I'm going to ask one more to see if there are any other questions coming from the floor, so to speak. You quote the biomarker p-tau181 as a progression marker, and is that just due to the availability of the data, or do you think that's a preferred biomarker over p-tau217?
I don't know. I just had this available, and that's plasma biomarkers. I also know that they have a higher number, if you like, for p-tau, and they say they are more predictive, 240 something, 241 or so. Yes. So for me, it was just data availability.
Yeah. Okay, that makes sense. All right. So I'm looking for questions in the chat. Are there any other questions for Valentina?
maybe I just put the proposal to whoever listens to me. So if you have polygenic risk scores, don't be surprised that with different methods and with different GWASs, you will have different value for the same person, right? What you need to do, what I always suggest people to do, think what you want to achieve. If you want to just publish paper and show general tendencies at polygenic risk score, predictive or non-predictive for whatever disease you're studying, that's fine to use just any of these methods, because for Alzheimer's disease, they will give similar results. But go for perhaps PRS-CS, it's easy to run, it's Bayesian approach, and the reviewers won't ask you questions. But if you really want to go to understand exact risk for these people, you need to think properly about study design and what exactly you are predicting. Then you might choose a genome-wide association study of appropriate age and appropriate clinical assessment of cases and controls rather than general dementia. So that's first. And second, in terms of methodology, again, if you're interested in a particular biological process, I don't know, set of microglial genes, then you basically focus on them only and calculate PRS for this set of SNPs or genes of your interest. That will be more precise rather than including everything into one big pot.
Yeah.
Or ask me questions. I'm happy to collaborate and explain more.
Yeah. So I think that's wisdom itself. And I think it does take the field away from this blanket
Yes.
risk of genes to specific pathways.
Yeah.
So I think that the next big advance I anticipate in Alzheimer's disease ascertainment is using proteomics pathways rather than clinical diagnoses.
Yes.
Do you think that will affect your use of PRS or not?
Yes. What I think so far, I'm modeling two variables, ApoE and the rest. What I think when we define these pathways, we can use a number of variables, 5 perhaps, based upon these proteomics or other processes, when we can, the model will be much more sensitive and much better predictive and individual. What's important is not overall p-value, overall area under the curve. What's important, what individual people have as a score, because these people will be perhaps given drugs to try or perhaps taken for clinical trials, etc. That was important. And for precision medicine, of course, every person counts, not the overall p-value.
Yeah, excellent, lovely. Well, that's I find that really exciting, actually. As I say, that would be the next big advance in my view. Okay, so unless I see another question in the chat, I'm going to Valentina, thank you very much indeed. Absolutely fascinating and competent as always. It's always good to hear you. So
yeah,
thank you very much indeed.
And thank you for inviting me to do the seminar here.
Okay, bye-bye.